DNA and protein synthesis. DNA directs the synthesis of proteins which carry out various cell functions. It could be the creation of enzymes or the development of muscle mass if you're increasing your muscle mass. Any kind of proteins that your body needs to function. DNA is responsible for their synthesis. DNA, however, remains in the nucleus. This green is the chromatin, and protein synthesis happens in the cytoplasm. And what we'll go through today is the transcription, which happens in the nucleus, which is DNA is transcribed by RNA, and the translation of RNA to protein in the cytoplasm. Those are the things we will cover today. This here is a cell membrane, the cytoplasm, the cell nucleus, and the chromatin inside the nucleus. Transcription is where DNA is transcribed by RNA, and we call that messenger RNA. Messenger RNA carries the DNA code out of the nucleus and into the cytoplasm. And translation, where the RNA is translated into protein, happens in the cytoplasm. Transfer RNA is involved with that as it interacts with messenger RNA to create the protein. So DNA in the nucleus is the code, is the information that is transcribed by messenger RNA, which interacts with transfer RNA in the cytoplasm to create the protein. We'll see that in the one of these next slides. Transcription happens when DNA partially unfolds aided by RNA polymerase. Notice that ASE is an enzyme. RNA polymerase is an enzyme. Messenger RNA and transfer RNA are both formed in the nucleus, and they're sent into the cytoplasm where the protein is synthesized. Here is a drawing representing that process. This here is the nuclear envelope. This is the cytoplasm of the cell outside the nuclear envelope. Here we have nuclear pores. Okay, this is the double helix DNA, and with the help of RNA polymerase, messenger RNA is made. And notice that its code is defined by the code that is on the DNA. We have the pointy adenine, the pointy indent thymine, the rounded cytosine, and the rounded indent guanine. They match up. The nucleotides match up. In messenger RNA and transfer RNA, uracil is a stand-in for thymine. And here we have the transfer RNA being made also inside the nucleus, and the transfer RNA has 3 nucleotides in a specific sequence attached to it. And both the transfer RNA and the messenger RNA all leave the nucleus and enter the cytoplasm of the cell. In the cytoplasm, we have 3 components working together: the messenger RNA, the transfer RNA, and the ribosome. The ribosome recognizes 3 codons, 3 nucleotides that are called a codon, at a time. Each codon is specific for a specific amino acid. Remember, amino acids join together to create the proteins. 9 anticodons then would be 3 amino acids. In the cytoplasm of the cell, transfer RNA, with the help of ATP, binds to the specific amino acids that match the codon that are with that transfer RNA. Here we have a pointy indent and a round, 3 nucleotides, and this is the polka dotted amino acid that matches those 3 nucleotides. The transfer RNA then is called the anticodon to the messenger RNA because those are the opposite base sequences to the messenger RNA. These amino acids line up and they have a polypeptide bond, which then translates into a specific protein at the endoplasmic reticulum. So translation is the transfer RNA and the messenger RNA translating into this protein sequence, and that happens on the endoplasmic reticulum where the ribosomes are located.